Hello and welcome back. So this is where we left off at the last video. Activation of B cells, activation of T cells. Here we kind of focus on where do the signals come from, but I really want to walk you through what MHC is and really help you get straight the difference between MHC1 and MHC2. So I'm going to show you some ugly, really gnarly pictures. Well, here we go. This is what I'm showing you. This is from a clever textbook or website. I don't even remember where I got this. What I like about it is how it shows the things locking together. So this slide is showing T cells being activated by a dendritic cell. So this is the mature, naive T cell. And it's important to note here, it will become important to note, this is a CD4 positive T cell. So this is going to be a helper T cell. And that means it has this CD4 co-receptor. We'll talk about that in a second. But it also means that what it's looking at is a protein showed to it by MHC class 2. So MHC is involved here. Whenever a T cell is looking at something, it's looking at something displayed by MHC. But in particular, if it's a CD4 positive cell, it's looking at MHC2. If it's going to be a CD8 positive cell, it's going to be looking at MHC1. That's a mnemonic thing I can show you here. This is the mnemonic device. You kind of take the numbers and pretend you're multiplying them, and they always come out to 8. So CD4 positive cell is going to work with MHC2, because if you multiply 4 by 2, you get 8. CD8 positive cell is going to work with MHC1, because if you multiply 8 by 1, you get 8. Just remember the rule of 8, and you'll be able to keep that straight, and that will rhyme. Right, so let's look at the pieces of this diagram. So again, this is a dendritic cell, and when I draw dendritic cells, they're really lumpy, and they have these projections, and they're like crazy amoebas, but realistically, under the microscope, they look like this. They just have a lot of little projections shooting out that are almost too small to see. So this is really what a dendritic cell is shaped like. What it's showing is the dendritic cell doing phagocytosis. So it's bringing an antigen into a vesicle. Okay, that's the idea. And it will fuse lysosomes with that vesicle to break down the antigen into parts. So into protein fragments, epitopes. And once the lysosomes have fused with this vesicle, this phagosome we call it, once they've fused, it becomes a phagolysosome. And so a dendritic cell, its whole job is to show things to T cells. So it has a system for taking proteins out of the phagolysosome, loading them onto MHC class 2, and then displaying that at the cell surface. So that's what this is. I hope you can see my mouse because I am indicating the yellow upside down U. I hope you can see my mouse. So that's supposed to be MHC. And the way it's drawn, you can't tell if it's MHC1 or MHC2, but we know it's MHC2 just because it's interacting with a CD4 positive cell. And let's keep going. So this is the epitope, which is usually part of a protein. The adaptive immune system really looks at proteins more than anything else. And then this is a T cell receptor. This is that super unique receptor that only this T cell has. And it's the only receptor this T cell has. So it's both unique and it's limiting. This T cell can't see very much. So it's looking at this peptide to find out, is this the peptide that matches its receptor? Does this match its receptor? That's what it's asking in this interaction. Almost always, the answer is no, because most antigens that end up in a human body are self-antigens, and there's no way a T cell should recognize them. But also, this receptor is randomly generated. There's no guarantee that it matches any antigen that it will ever see. So that's the T cell receptor, and then this is the CD4 co-receptor. And the idea is this sticks out of the T cell, and it grabs the MHC and stabilizes it. So it kind of aligns it correctly with the T cell receptor and makes sure that T cell receptor is able to see the peptide. So this T cell receptor is made of 2 proteins. MHC is made of some proteins. I don't remember. CD4 is a protein. So, okay, so you've seen all these parts. What I want to point out to you is when MHC2 is used, it isn't just because it's looking at a CD4 cell. There's something specific about it. It is used to display the contents of phagolysosomes, generally speaking. So when a cell brings something in a vesicle and breaks it into pieces and displays it, it has to use MHC2 to do that. And so CD4 positive cells are the only ones that can see proteins that came from phagolysosomes because they're the only ones that can see MHC class 2. Okay? So there's a lot you can get out of this, and I definitely suggest drawing these diagrams and including everything that's named here. Draw it. So this cell pulling something in through phagocytosis, breaking it down, presenting it, that means it's going to be presented on MHC2. That means it's being presented to a CD4 positive T cell and its T cell receptor. And that means the CD4 protein coming out of the T cell is grabbing the MHC and stabilizing it. Okay, that's what we see in this. Remember that, because when we look at MHC1, it will look similar at first, but it will be a very different situation. So, okay, again, T cell activation. The dendritic cell presents antigens to the mature naive CD4 positive T cell. I think I wrote things here. So yeah, the dendritic cell runs around doing this interaction with as many T cells as it can, and again, most don't recognize whatever it has. It has to use MHC2, CD4 co-receptor, and if this T cell receptor does recognize this epitope, well, that's wild. That means that this dendritic cell is the hero that has found the correct lymphocyte. This lymphocyte is going to help save the life of this person, because this is the one that can respond to whatever is infecting the body. So if that's true, then this cell is going to go through clonal expansion, and its daughters will go through differentiation. Okay? Here is a lymph node. This is a super close-up of a cross-section of a lymph node, and all these little dots are the nuclei of lymphocytes. This is showing where T cells hang out. And I'm not good enough at the histology to point out dendritic cells, but there are dendritic cells and macrophages in here. And you can't tell by looking at a lymphocyte under a light microscope. You can't tell if it's CD4, CD8, or even a B cell, except that T cells hang out here, B cells tend to hang out in a different area of a lymph node. So part of why I show you this is I just want you to remember that all these lymphocytes might be different. These might all be T cells with different T cell receptors. And if we want to trigger an immune response, we have to find the right one that can actually see the protein coming from the pathogen. And so it is very much finding a needle in a haystack. It is finding the right lymphocyte when they all look the same. And the way they do it is they use this system, this MHC, and they just do this interaction between MHC displaying the protein to a T cell receptor stabilized by a CD4 or CD8 as fast as they possibly can to as many T cells as they possibly can. Okay. That was MHC2. Let's look at MHC1. This is different now. Here, this is a cytotoxic T cell. So this is not a cell that's being activated. It's an effector cell. So this is one of the daughter cells of the cell that got activated. And so this is doing its effector function. And this cell, unfortunately, is infected with something. So it's got a virus, and the virus is making mRNA, and it's being translated into proteins, and they're being broken down and displayed on MHC1. The virus doesn't want this to happen, but this is what all of our cells do all the time. They just grab random proteins from the cytoplasm, put them on MHC1, and display them. Just for this situation. This cell doesn't have any way of knowing it's infected by a virus, but it does have a way of showing a cytotoxic T cell whether or not it's infected by a virus, because it shows random proteins, and viral proteins will be very abundant during an infection like this. And so if this cytotoxic T cell is responding to an infection of this virus, it's going to recognize this peptide. And it's looking, again, using its T cell receptor. And it's stabilizing this interaction with its CD8. And its job is, if it recognizes this peptide, it knows this cell is infected with whatever activated this cell's ancestor. So something abundant enough in the body that a T cell all got activated. And here it is. So this cytotoxic T cell's job now is to kill this whole cell, is to just send it signals for apoptosis, programmed cell death. And when this cell dies, it stops being a virion factory. It can't make any new viruses. This is what stops a viral infection. So remember, antibodies can prevent attachment, but cytotoxic T cells prevent replication, period. And once attachment has happened, the virion gets in and turns the cell into a virion factory. And the only really good way we have of stopping that is this. The cytotoxic T cell identifies it and kills it. So there's that. So here it all is. Here it all is. Okay, cool. That's the mnemonic device. So those were two situations where you'll see an interaction between a T cell and a cell displaying a protein. There are 5 of these situations where MHC is displays a protein, an antigen, to a T cell using its T cell receptor. So these are the critical situations, and if you get all these situations, you can draw a diagram of the whole adaptive immune response. And that's kind of awesome. So in each of these, I'm going to write 5 of them, and the first thing you're going to see is going to be what cell shows something to what T cell, generally, using which type of MHC, and then there's going to be an arrow. And so if the T cell actually recognizes the protein, what will happen? So let's look at the first one. This is going to involve MHC1, and this is a dendritic cell showing proteins to a mature but naive CD8 positive T lymphocyte. So this is trying to activate the right lymphocyte. The dendritic cell has all these antigens, and it's showing them to lymphocytes. And dendritic cells have both MHC2 and MHC1 because they need to be able to activate CD8 positive cells and CD4 positive cells. So they're not limited to what I showed you on the previous slide. So this dendritic cell can show proteins using MHC1 to the mature naive CD8 positive T lymphocyte. And if it recognizes that protein, that antigen, that epitope, then it will go through activation, and its daughter cells will become cytotoxic T cells or CD8 positive memory cells. Okay, so that's the first one. There are 4 more. And I understand you need to go through this very slowly. Very slowly. Like, I can say this fast, but you really need to slow it down. So I'm not going to explain the 5 situations here. unless I see something that needs to be explained, because really you need to work through this, right? You need to build your own understanding, and this is all like word soup, and you need to untangle it. So I'll get out of your way. This is what I showed on the last slide. And notice it says typical nucleated host cell shows cytoplasmic proteins to the CD8 positive cytotoxic T cell. So if we go back here, this is the CD8 positive cytotoxic T cell. Well, this is an infected cell, but this cell would be doing this whether it was infected or not. It's always doing it just in case it might be infected. That's how we find the cells that are infected. They're all doing this, and most of them are displaying host proteins. They're displaying their own proteins. And cytotoxic T cells can't recognize host proteins. So that's situation 2. Situation 3 is where this is going to be activation of a CD4 positive T cell. Now this is effector function, so we're not looking at activation anymore. This again is effector function, but it's going to use MHC2. And the macrophage is showing its phagosome contents to a helper T cell type 1. And this is where the macrophage can send it activation signals. Now, I think that would make sense to you. The last one is the wildest one. It's going to be a B cell talking to a T cell. Because a B cell is, circulating in the blood or the lymph, and it finds its antigen, but it doesn't know whether its antigen is important. It doesn't know whether it's important. It just knows it found its antigen, so it has to go find a type 2 helper T cell and ask it, do you recognize this? And if it does, they're both important cells in an immune response. They're just like high-fiving, like, yes, we are the critical cells in this immune response. I'm the helper T cell, and you're the B cell that's going to give rise to all the antibodies. And so that B cell will activate. Just think how special a lymphocyte feels when it activates. It is a hero already when it activates. And all the other lymphocytes looking at it like super jealous. So, during a Zoom session once, I drew out the adaptive immune system in all 5 of those situations, and you can too, and you should too. Notice I'm always drawing like 3 lymphocytes. These are B lymphocytes, T lymphocytes. I'm just always drawing 3 to remind myself that they have unique lymphocyte receptors, and one of them will get activated, or maybe 2 of them out of 1 million will get activated during an infection. So I highly recommend you drawing these things out if you want to understand them. Now this is yet another of the very long videos from the immune system, but I hope you can see this has all just been on the topic of MHC and how it's used. And I appreciate your patience with this, because this is a gnarly topic. But if you understand it, you understand the adaptive immune system. So that's a thing not everybody can say. Well, thanks again for your attention to this. We're going to shift to other topics that probably are going to be smaller and more bite-sized. The adaptive immune system is this big, complicated thing, and nothing else we're going to look at is quite that complicated. So I'll see you soon.